Thank you for inviting us to speak at the 16th Annual Emiquan Science Symposium. This is a wonderful annual tradition, and we are very pleased to be part of this conference. My name is Dean Bobo. I'm a PhD student at Columbia University. Today I'll be presenting alongside Mike Lemke from the University of Illinois Springfield and Rob DeSalle from the American Museum of Natural History. This talk focuses on Thompson Lake. And while the history of the lake is a fascinating success story involving river ecology, land use management, and restoration, our work attempted to characterize the most abundant life forms in the lake, the microorganisms, and more specifically for our story today, the bacteria. Though microorganisms has always been a part of human life, much more attention has been given to characterizing the human microbiome over the last decade. We continue to learn more about the many critical roles that microbes play in human health. Many pathologies and physiologies ranging from stomach ulcers to human emotions involve microbial communities, and often a simple change in diet or a prescribed probiotic can help manage our health for the better. This figure shows that only 6 of the 89 commonly recognized bacterial phyla are common in the human body, and different parts of the body have different proportions. The human body to a microbe is indeed an ecosystem. What we would like to present today are the findings about the open water Thompson Lake habitat and the Emiquan ecosystem. Just as with the description of the human microbiome, we hope that understanding the lake microbiome will lead to a deeper understanding of the ecosystem and ways to make the system healthy in an ecological sense. Our primary site was Thompson Lake in the Emiquan Preserve, though sampling of other water bodies was done for comparison and to gain perspective. Here we present results from our frequent bi-weekly sampling of the surface water of Thompson Lake at 3 fixed sampling points shown here, T1, T2, and T3. Sampling ran from about March to November for every year starting in 2008 and going through 2016. While sampling, we recorded common limnological measurements listed in this slide. Our chemical parameters were measured from whole or filtered water. Pelagic organisms were captured on a 0.2 micron pore membrane filter, and dissolved nutrients were measured from the filtrate. DNA was extracted and used for 2 methods of community analysis: ELISA and 16S ribosomal RNA sequencing. ELISA stands for Automated Ribosomal Intergenic Spacer Analysis. I'll explain this briefly. Each species of bacteria has spaced DNA between the 16S and 23S genes. The spacer DNA's length is different for most species. Thus, if a water sample is filtered, the DNA is extracted, and the spacer lengths are determined, the number of sequences of the same length coupled with the presence or absence of that species' DNA allows a similarity matrix to be created. Thus, in our graphs, each point represents many species, and the closer the points are to each other, the more similar the samples are. When we began in 2008, Thompson Lake was essentially a flooded cornfield. Each year, the lake developed different characteristics, and we began noticing changes in the microbial community by season. We also documented a very big shift in the microbial community in the middle of 2010. The patterns we found are fascinating, and of course, as curious scientists, we wanted to know which taxa were involved. Our current findings address this objective. What are the species of bacteria in Thompson Lake? With this knowledge, we thought we could start to understand some of the functions and interactions, and we might be able to explain some of the ELISA patterns. Thus, we did Illumina sequencing of the 16S ribosomal RNA gene. 16S ribosomal RNA is a component of the prokaryotic ribosome, the molecular machinery responsible for protein synthesis. The genes encoding 16S RNA evolve very slowly, which makes them excellent targets for identifying taxa and reconstructing evolutionary histories of bacteria. What we found at the phylum level was that Thompson Lake shares 4 of the 6 most common bacterial phyla found in the human body. And perhaps most notably, the proportion of phyla at Thompson are similar to the human gastrointestinal tract. We're not trying to say Thompson Lake is a human, but what we are saying is that many of the decompositional processes that go into digesting food for energy exist in organisms as well as ecosystems, and this is a good thing. There are a lot of data for nearly a decade of sampling, but let's focus on years 2008, 2010, and 2012 for a slice in time and see what occurs by season. The first thing we notice is we see the four main phyla change proportion by season, with mainly the relative abundance of Cyanobacteria and Bacteroidetes being the most variable. Over the 5-year time period shown, we see the emergence of another phyla, the Planctomycetes. We see these results to be very exciting when they were initially described as a eukaryotic species, but Planctomycetes are indeed bacteria, and they have a wide distribution in aquatic and terrestrial habitats, and they adapt to both aerobic and anaerobic conditions. What interested us most in the case of Thompson Lake was that more recently, members of this phyla have been described to fill in a considerable missing part of the nitrogen cycle. That is, the anaerobic ammonium oxidation. The fact that this phyla became prevalent after the nitrogen-poor waters of Thompson Lake received nitrogen from nitrogen-fixing cyanobacteria gives great credibility to the use of phylogenetics to trace lake conditions. Looking again at ERISA data for most of the sampling period plotted in non-metric two-dimensional space, we found that communities for 2008 to mid-2010, which are orange, yellow, and red, differ from late 2010 to 2013, which are red, green, blue, and purple. Something happened about 2.5 years into the restoration that changed the microbial community. First, there was a phosphorus spike in 2008. We will see that this helped to get one group of autotrophic nitrogen-fixing cyanobacteria an advantage in the open water.
Hi everyone, this is Mike Lemke. It's the halfway mark of the presentation and I'm the designated relief speaker, so let's continue. We are in the middle of making a case for a microbial community shift in mid-2010 and laying out where the cyanobacteria fit in and why. This graph shows the 16S data for the families and orders we found in the Cyanobacteria phylum. These data confirm work done by microscopy that a member of the Nostocacae family, the Aphanizomenon, bloomed initially. Note that Aphanizomenon is shown in dark red bars on the left of the graph. But in the later part of 2010, the order Cyanobacteriales, or the Cynococcus cyanobacteria, which are colored in blue, became the dominant cyanobacteria. Here is a picture of the Aphanizomenon bloom. At first glance, it is hard to believe that tiny bacteria are making these large green clumps, and these clumps can regulate, to some extent, their buoyancy and thus place in the water column. And it is this clumping, incidentally, that caused variable water clarity early in the Thompson Lake restoration. The exact conditions that set off cyanobacterial blooms are not well defined, but Aphanizomenon really likes phosphorus, and it appeared to take advantage of that first phosphorus pulse we showed you a few slides ago. But also, Aphanizomenon likes phosphorus so much that it produces a compound called cylindrospermopsin, a compound which helps to scavenge phosphorus from other organisms by inducing alkaline phosphatase activity. Alkaline phosphatase hydrolytically releases phosphate from organic compounds. Thus, with its ability to control buoyancy, produce colonies, and fix nitrogen, Aphanizomenon took over the lake early in restoration. My rough calculations indicate that its own dying biomass brought in about 1/4 ton of nitrogen to the lake, making nitrogen no longer a limiting nutrient, and by 2010, most soluble phosphate was tied up in biomass and thus less available. As nutrient conditions changed, Aphanizomenon gave up its dominance and Cynnococcus took over, at least in the Cyanobacteria part of the story. It should be noted that another process separate from nutrient competition could be at work here. The recent literature suggests that a cyanobacterial virus or cyanophage could also play a part in decimating a population. We simply do not know at this point. Besides a shift in cyanobacterial species, another very important change in the lake occurred in 2010. Less light and thus less energy going to greater depths in the lake. With the change in mid-2010, variable water clarity shown in the black circles went to shallow clarity of about 0.5 meters. So we're talking about here where we have light going down to about 2 meters at times and other times not nearly that deep. After 2010, we're sitting here at about 0.5. The Cynnococcus cyanobacteria that replace the Aphanizomenon are very small. They don't make the clumps like Aphanizomenon and therefore likely did not induce the decreased water clarity by itself. Our colleagues at the University of Moringa did the microscopic analysis of the algae. In mid-2010, the larger eukaryotic algae, such as diatoms, green algae, cryptophytes, and even the rhapidophytes, took over much of the microautotrophic role in the lake. Now let's move from looking at longer-term trends that occurred over several years to trends that are taking place on an annual basis. Many of us are well aware of shifts in algae populations, microcrustaceans, and other aquatic organisms and how one group affects the other. Let's see what we can learn from the bacterial seasonal patterns. Since nearly 1/2 of the bacteria in Thompson Lake were Bacteroidetes, we began our investigation of seasonality there. This phylum holds many of the carbon decomposers. The most abundant natural polymers in the world, cellulose and chitin, have members of the Cytophagales and the Chitophagales that hydrolytically decompose these polymers. These groups are the orange and purple colors in this stacked bar graph. And we see that they show up each year that we did our sampling and analysis, but to varying degrees throughout the year. Flavobacteria in the Actinobacterialis, the yellow and pink colors, favor breaking down plant carbon compounds, including simple and oligosaccharide sugars. Reduced carbon is energy to the heterotrophic bacteria cell, and each population has adapted an opportunistic way of acquiring and assimilating that carbon. We speculate here that the bacterial species abundance follows annual carbon availability, likely keying on algoxidizers in the spring, more chitin later in the year as microcrustacean deaths accumulate, and use of plants in a range of decompositional states that are occurring in Thompson Lake to sustain these Bacteroidetes populations. What we have presented today are findings about the open water Thompson Lake habitat in the Amaquan ecosystem. We covered the shift over the sample period from the microcell autotrophic point of view, explained which phyla were common and which showed up and were of great interest from a nitrogen cycling point of view, and have presented a working hypothesis about the seasonality of the bacteria that are the digesters of the lake carbon. Just as with the description of the human microbiome, we hope that understanding the lake microbiome will lead to a deeper understanding of the ecosystem and ways to make the system healthy in an ecological sense. Will we be feeding the lake more yogurt or other probiotics? Well, likely not yogurt, but we could consider useful inoculations, like those from the Illinois River, are possible. Will Thompson Lake need to be given probiotics? Possibly. An influx of labile carbon at what is considered a timely part of the year could shift community outcomes dramatically. But we are still in the information assimilation stage of the work, so stay tuned for more developments. That is the end of our presentation, and I thank you so much for your attention. We would like to thank also all of the people that made this research possible, which include many students, faculty, and staff from the University of Illinois Springfield, scientists at the University of Moringa, the American Museum of Natural History, and Columbia University, and also a special thanks to those at UIUC who helped with DNA extraction and the ERISA work, and of course a special thanks to the Nature Conservancy who have persisted in support of work for these many years. We appreciate funding always, and especially from the sources listed. At this time, we stand by to answer questions. Thanks again.